Good afternoon, students. So today in my lecture, I will discuss about the structure of prokaryotic cells, and then from chapter 2, we will discuss about the biological classification, in which we will discuss about the early systems of classification. Again, I will discuss about the R.H. Whittaker's five kingdom system of classification, and everything about the classification of kingdom Monera. So we will discuss this topic from chapter 2, and from chapter 2 I will discuss about the structure of a prokaryotic cell. So afterwards I will discuss about this assignment which I sent you on Wednesday. So today I will discuss about this worksheet. So let's first start with the biological classification. Uh, there is a word classification in bio. It is very important to classify or to group, to provide a particular category to the organism, because it is very difficult to study each and every organism at individual level. So what we do, on the basis of certain characteristics, we classify the organisms into common categories. We group together them in a common category. There were some early scientists, those who tried to classify the organisms, like Aristotle. Aristotle was the earliest to attempt a more scientific basis for classification of organisms. So he classified plants on the basis of the length, size of the plants, like trees, shrubs, and herbs. And in the animals, he classified the organism on the basis of presence of RBCs. So that was not the best way to classify the organism, but anyhow, that was the time when people started classifying the organism into different categories. Then there were Linnaeus, Carlos Linnaeus, who is known as the father of taxonomy. Taxonomy is the branch of science which deals with the identification. Nomenclature means giving name to the organism, and based on certain characteristics, classifying into a particular category. We call it as taxonomy. So, Carlos Linnaeus, who is known as the father of taxonomy, father of classification, father of nomenclature, he introduced two kingdom system of classification, that is kingdom Plantae and kingdom Animalia. But there were certain drawbacks of this classification, that the Carlos Linnaeus included all the and many prokaryotes and eukaryotes into one category. Like under plants, he classified fungi, gymnosperms, angiosperms, mosses, and bacteria, cyanobacteria, classified under one category. You know that prokaryotes, bacteria and cyanobacteria are the prokaryotes, and the fungi, mosses, gymnosperms are the eukaryotes. So that was not the valid method to classify the organisms. Even you know that fungi, which are saprophytes, although they have cell wall, but they are saprophytes, and the cell wall is made up of chitin. So they are not similar to plants. So he classified all such organisms into common category. So it was just based on the cell wall. As we know that in prokaryotes there is a cell wall. In case of fungus there is cell wall. In case of plants there is cell wall. But there is a difference in the composition of cell wall. Then he also classified unicellular and multicellular in same group. Like Chlamydomonas is a unicellular algae and Spirogyra is multicellular. So he placed under unicellular and multicellular in the same category. So he could not classify the heterotrophic fungi and autotrophic green plants into different categories. So afterwards, the five kingdom system of classification came in 1969, when R.H. Whittaker proposed five kingdom system of classification. So last week we discussed that on the basis of certain characteristics, that is cell wall, nuclear membrane, then cellularity, then modes of nutrition. On that basis, R.H. Whittaker classified the organisms into five categories, except viruses and lichens. I discussed that why he could not classify viruses and lichens into the five kingdom system of classification. So if you will look at the Monera, which is the first kingdom, here The cell wall is non-cellular, made up of these peptidoglycans, a complex sugar. We also call it as mucopolysaccharide. And the nuclear membrane, here it is absent. We discussed last time. Body organization, it's a cellular level of organization. Mode of nutrition, it includes autotrophs, heterotrophs, and even some parasites too. So there is a, what you can say, there is a dynamic mode of nutrition. There is a diverse type of nutritions in the kingdom Monera. There are autotrophs also, and there are heterotrophs also. If you will look at the protista, here there are unicellular eukaryotes. Rest all the characters are same like as multicellular organisms. The thing is that here the organisms are unicellular, and again here there are diverse groups which show different types of nutrition, like these include autotrophs as well as heterotrophs. Fungi are purely heterotrophs. They are multicellular except in case of yeast, which is unicellular. But rest all the characters are same like the eukaryotes. They have cell wall which is without cellulose. It is made up of chitin. In plant, cell wall is there, but in animalia group the cell wall is absent. So you should know the basic criteria of classification of RH Whittaker, a five kingdom system of classification. Here some merits of RH Whittaker classification are given, like, so it was more scientific and natural. Prokaryotes and eukaryotes were classified into different groups. Unicellular and multicellular organisms were categorized into different categories. So more evolutionary relationship was followed, means um, the kingdom, like the kingdom Plantae and Animalia, show the phylogeny of different lifestyles in the five kingdom classification. They are more homogeneous group than the two kingdom classification, means one type of organism you will find in one category. So he could classify the organism on the basis of their ancestral relationship, on the basis of their cellular characteristics, on the basis of nutrition. He could classify the organisms in a better way. But still there were certain loopholes, there were certain disadvantages of the RH Whittaker's classification. Like here you will see you can see the system of classification drawbacks with reference to the lower forms of life, like the kingdom of Monerans and the Protestants include diverse heterogeneous forms of life. Heterogeneous means there are different forms of life, like in kingdom Protesta there are Protestant algae which are plant like. Then there are Protestant slime molds which are fungus like. There are protozoans in kingdom Protesta which are animal like. So the lower groups are heterogeneous, having both autotrophic and heterotrophic nutrition. Even there is a difference in the cell wall. In the kingdom Protesta there is a group called euglenoids. They have cell wall like structure. We call it as pellicle. In lower group, in some cases there is cell wall but in some cases there is no cell wall. So there is a diverse classification even in a single group. There is so many differences. You will find the unicellular algae like volvox and chlamydomonas, which should be classified under kingdom Protista, were earlier classified into kingdom Plantae. Okay, so you should know. Then the general organization of slime molds are completely different from the members of the protists, which are fungus like. He could not classify viruses and lichens. So there were certain demerits of the RH Whittaker classification, but equally there were so many merits of the RH Whittaker's classification which you should know, which is very very important. So here just a diagrammatic representation of the five kingdom system of classification, show the diversification, that how from one group to another groups would look. Here first Monera came into existence, then Protestant, from Protestant the fungi and Animalia came into existence. Here from you can see the plants are forming. Here it shows kingdom Monera, here it shows Protesta. Then on the basis of the nutrition, here it is written photosynthesis, so plants show photosynthesis. Fungi are saprophytes, in which the digestion of food takes place outside the body, and then they absorb the digested food. We call them as saprophytes. That is why here it is mentioned absorption. Then Ingestion, Animalia group shows the holozoic nutrition which ingest the food, solid food. So that comes under the Animalia. So ingestion, photosynthesis, absorption, so show different types of nutrition in different groups. Then there are three domains also. Now before kingdom, we also classify the organism into three domains, which I have not discussed here, I have not mentioned here. You should know there are three domains. One is the prokarya, in which we classify the true bacteria, to which we call eubacteria, and other. And second one is the archaea, under which we classify the archaebacteria or the primitive bacteria. And third one is the eukarya. Under eukarya we classify the organisms like fungus, plants and animals, and even the protists. So there are three domains which you should know. One is the prokarya, second is the archaea, and third one is the eukarya. So you can search on the internet or you can find out from your reference book that there are three domains. One is the prokarya, under which there are eubacteria or true bacteria. Second one is the archaea, in which there are the primitive bacteria. And third one is the eukarya. One is the prokarya, then archaea, then eukarya. Under eukarya there are all eukaryotes like protists, fungus, plants and animals. So there are three domains under the classification, under which there are 5 kingdoms. Now I am discussing about the kingdom Monera. Last time we discussed based on the structure. If I can show you the structure, here is the structure of the bacteria. Here you can see in bacteria there is no well-defined nuclear membrane. We call it as nucleoid. Okay, and there are no membrane-bound cell organelles like mitochondria, Golgi apparatus, lysosomes, vacuoles, ER, etc. All such membrane-bound structures are absent. So monerans are considered as the most primitive group of organisms. So there, even it is estimated that a single drop of water may contain 50 billion bacteria. So they are very much versatile in habitat. They are found everywhere in the world, even in the hot thermal vents and even in the colder regions, extreme colder regions, you will find the monerans. So Monera includes the eubacteria or true bacteria, then there are archaebacteria, then mycoplasmas, then actinomycetes and some photosynthetic cyanobacteria, or to which we also call blue-green algae. So these are the groups of kingdom Monera. So monerans, as I told you, they show diverse metabolic nature, means they include autotrophs as well as the saprophytes as well as the parasites, heterotrophic nutrition. They show photoautotropic nutrition as well as chemoautotropic, means if they are autotrophs they can fix the inorganic compounds in the presence of light as well as by using the chemical energy. They do not have nuclear membrane, they have single chromosome. Okay, they may have additional ring of the DNA, to which we call plasmid. I will discuss it later on in the topic the structure of prokaryotic cell. Okay, so based on the characteristics, all this, even on the basis of the shape, there are different types of bacteria. Like here the rounded shape is the coccus bacteria and the rod shaped is the bacillus, then spiral shaped it's the spirilla, and the comma shaped is the vibrio bacteria. I can give you the example like lactobacillus, which helps in the formation of curd. It's a coccus bacteria. Oh, sorry, bacillus bacteria, lactobacillus, it's a rod shaped. If we talk about the streptococcus, which is used for making the antibiotics, it's a coccus bacteria, round shaped. Vibrio cholerae, which cause cholera, it belongs to Vibrio category. So there are different types of bacteria on the basis of shape. Even you can classify the different types of bacteria on the basis of the number of flagella and the presence of flagella. So that's what I'm not discussing, and now I'm just discussing the part from the NCRT or little bit out from the NCRT. So as I told you, kingdom Monera has diverse classification. We can classify it into two main groups, like into two domains. One is the prokarya and another is the archaea. In prokarya there is a eubacteria. In the archaea there are primitive bacteria. Archaebacteria are the primitive bacteria having different cell wall composition. Due to their different cell wall composition as compared to the other eubacteria, they can live in the extreme conditions. We call them as extremophiles. They do not have peptidoglycan in their cell wall, and their cell wall is made up of a chemical known as pseudomurine or non-cellulosic polysaccharide. So this is the condition by which they can live in the extreme condition. Extreme means they can live in the salty areas, they can live in the thermal vents, they can live in the marshy areas, they can live inside the body of the ruminants like cows and buffaloes. So there are, based on the habitat, there are three types of the uh, you can say archaebacteria. One is the methanogen, another is the halophile, one is the thermoacidophiles. Methanogens are those uh, which live in the marshy areas or in the stomach of the uh, ruminants like cows and buffaloes, and they produce methane. So we call them as methanogens. Those live in the salty area, we call them as halophiles. Those live in the uh, high or low temperature or in the thermal vents, we call them as thermoacidophiles. They have such enzymes which can work at high temperature. Okay, so you should know about the classification of the archaebacteria. And archaebacteria are the living fossils. They were the first who came into existence on the earth. So you should know that archaebacteria are known as the living fossils. They were the first who actually uh, came on existence on the earth. So then the second group is the eubacteria, to which we call true bacteria. They have rigid cell wall. They may have flagella for the motility. So they include diverse groups. They include autotrophs as well as the heterotrophs. Like under the autotrophs, there are cyanobacteria. The example is the nostoc and anabina. In such groups, if you will see in the book, you will find there is a diagram of nostoc and anabina in which there is a specialized cells called the heterocysts which help in the nitrogen fixation. Please note down, please check your NCERT book. You will find that there are two examples in the book in second chapter, that is nostoc and anabina. They have specialized cells for fixing the nitrogen, to which we call heterocysts. They live in the colonies, and all the cells of the nostoc or the anabina, they are colonized togetherly by a gelatinous sheath. So whole colony is surrounded by the gelatinous sheath. So these are very common in the ponds or even in the paddy fields. You can see the green colored algae. We call them as cyanobacteria. So these are the autotrophs. So they have a chlorophyll A, like similar to the green plants. So from here you can also judge that how the plants were evolved from the prokaryotes, because plants have chlorophyll also, same chlorophyll A. Prokaryotes too have, so it shows the ancestral relationship also, which you should know. Then there are certain chemosynthetic autotrophs which by using the chemical energy can form their food, or they can use that energy. Then there are heterotrophic bacteria which have diverse role in the uh, environment, like they have some useful aspects also as well as they have harmful aspects also. Like if you will talk about the uh, in case of the useful aspects, if you talk, then there are certain bacteria which helps in formation of antibiotics or the curd, but you will find certain bacteria which cause diseases. So there is a great economical importance of the heterotrophic bacteria, so which you can judge from this. Okay, then if we talk about the reproduction in bacteria, they generally reproduce by spore formation or through the binary fission. But in some cases, if I can show you the diagram here, it is the conjugation between the two bacteria in which the one bacteria donates its DNA to the another bacteria. So there is a sharing of the genetic material. We call it as conjugation. So this is very common type of sexual reproduction in certain bacteria. It's a primitive type of reproduction. So you should know about it. Okay. So then there is another topic which you should know, that is the mycoplasma. This is the another group of the prokaryotes. Mycoplasma are the smallest known organisms. Okay, they do not have cell wall. Please note down the characteristic. This is very important question. 2 years back this question came in NEET. So you should know that mycoplasma are the organism without cell wall. They are the smallest known organism. They can survive without oxygen, means can live in the anaerobic environment, and many of them are pathogens in nature. They are pathogenic in animals and plants. So mycoplasma, then heterotrophic bacteria, autotrophic bacteria, bacteria, then archaebacteria. Archaebacteria have methanogens, halophiles, thermoacidophiles. So you should know about the classification and their examples. Then types of bacteria on the basis of shape, there are general characteristics. Okay, then RH Whittaker's classification, merits, demerits, then two system classification, Aristotle's classification. So all you should know in the detail. So that's all about the biological classification. In my next video I will discuss about the structure of the prokaryotic cell. Anyway, so we'll discuss maybe later on about the structure of prokaryotic cell. Thank you.